RNA is synthesized by DNA by a process called transcription. So when it is acting as non-genetic RNA, it will always be single-stranded. mRNA passes on the genetic message to tRNA, from tRNA to rRNA, and that process is called translation. Hello everyone. Welcome back to session 5 of this important chapter called Molecular Basis of Inheritance. What did we study in the last session? We studied about DNA, then DNA replication, what are the features of DNA. In today's session, we are going to study about a very important experiment, that is the experiment for semi-conservative nature of DNA replication. In the last session, we studied about the semi-conservative method of DNA replication, and the experimental proof for the semi-conservative method of DNA replication was given by Matthew in 1958. It was performed by Matthew C. in the year 1958. You can see this scientist, he is none other than Matthew. So what is this experiment all about? This is an experiment to prove that DNA undergoes semi-conservative DNA replication. So what did they do? They grew E. coli, Escherichia coli, in a medium containing 15 NH4Cl, that is 15 ammonium chloride, where 15N is the heavy isotope of nitrogen. As the only nitrogen source of many generations, the result was that 15N was incorporated into newly synthesized DNA. How was it possible for the incorporation of 15N into the newly synthesized DNA when it was subjected to a medium containing 15NH4Cl, ammonium chloride? So this heavy DNA molecule could be distinguished from the normal DNA by centrifugation. How was it possible for differentiating the newly synthesized DNA having 15N and the old DNA? So this heavy DNA molecule could be distinguished from the normal DNA by centrifugation in a cesium chloride density gradient. So what did Matthew do in this experiment in 1958? He grew E. coli in a medium containing, that is, heavy isotope, that is 15NH4Cl, as the only nitrogen source of many generations. So the result was that, what was it? That is, the 15N, which is the heavy isotope of nitrogen, was incorporated into the newly synthesized DNA. So this heavy DNA molecule could be distinguished from the newly synthesized by centrifugation in cesium chloride density gradient. So they then transferred the cells into a medium with normal, that is 14NH4Cl. Later on, what did they do? They transferred the cells, that is E. coli cells, into a medium with normal 14NH4Cl and took samples of various time intervals as the cells multiplied and extracted the DNA that remained as double-stranded helices. See, listen carefully. What did they do? They subjected the E. coli cells in a medium of heavy nitrogen isotope called 15NH4Cl. Then they were able to incorporate what? 15N in the newly synthesized DNA cells. Then they transferred the cells into a medium with normal 14NH4Cl and took samples of various time intervals as the cells multiplied and extracted the DNA that remained as double-stranded helices. So the various samples were separated independently on cesium chloride gradients to measure the densities of DNA. Why was it passed on to the cesium chloride density gradient? To measure the densities of DNA when it was transferred to a normal, that is 14 ammonium chloride nitrogen medium, right? So with all this experiment, they were able to prove that that the DNA that was extracted from the culture from one generation after the transfer, thus the DNA that was extracted from the culture one generation after the transfer from 15N to 14N medium after 20 minutes had a hybrid. What do you mean by hybrid? Had a mixture or intermediate density. And DNA extracted from the culture after another generation, after 40 minutes, was composed of equal amounts of this hybrid DNA and of light DNA. So there was a mixture of hybrid DNA as well as light DNA when it was subjected from one transfer to the other transfer. So with this, he was able to prove that the semi-conservative method of DNA replication. So moving on to the next very important concept, ribonucleic acid, RNA. In the last session, we have studied about DNA. RNA is nothing but ribonucleic acid. Ribonucleic acid. DNA was deoxyribonucleic acid. Right? Now, previous sessions we have studied that DNA exists only as a double-stranded structure and always acts as genetic material and has 4 nitrogenous bases: adenine, guanine, cytosine and thymine. Then what is DNA? DNA and RNA are both nucleic acids found in the nucleus of a cell showing acidic property. But as I said, RNA exists in 2 forms. One as genetic RNA, the other as non-genetic RNA. Non-genetic RNA. So, DNA always exists in one form as genetic RNA. Always its job is to act as genetic material. But in case of RNA exists in 2 forms. One is called genetic RNA, the other one is called non-genetic RNA. So, what do you mean by genetic RNA? For example, like some viruses, RNA acts as a genetic material. Then, like for example, in some viruses, it is the genetic material. It is the genetic material. And it is double-stranded. That is very, very important. Only when it acts as a genetic material, it will be double-stranded. If not, it will be always single-stranded. So, in case of non-genetic RNA, it is single-stranded. It is single-stranded. Here it is double-stranded. Here it is double-stranded. Here it is single-stranded. And the very function of non-genetic RNA is what? Synthesize protein. That is protein synthesis. Here you can make out that it is a single-stranded RNA. So, it is a single-stranded nucleic acid found in the nuclear material of viruses and in the nucleus and cytoplasm of eukaryotes. It is a polymer of nucleotides with 5'3' strand. The RNA is synthesized by the DNA by a process called transcription. So, RNA is synthesized by DNA by a process called transcription. So, RNA will have 3 very important components. Chemical components or chemical composition. That is, it is having, even RNA and DNA both are having 5 carbon sugars, that is pentose sugar, but the only difference is what? There one oxygen atom is less. So, the ribose sugar is pentose sugar. There also deoxyribose sugar. Here it is ribose sugar. Both are pentose sugars. Here it is ribose sugar. Then the 3 nitrogenous bases are A, G, C and U. U stands for uracil. The only difference between DNA and RNA with respect to chemical composition is there it is deoxyribose sugar, here it is ribose sugar. The thymine of DNA is replaced by uracil of RNA. Right, and it is also having a phosphoric acid. So that is about the RNA. So, nitrogenous bases, as I said, the purines are adenine and guanine. The pyrimidines are cytosine and uracil. So, in RNA, In RNA, purines are adenine and guanine. Adenine and guanine. Pyrimidines are cytosine and uracil. So here it is uracil. Very, very important. So in DNA it is thymine, but in RNA it is uracil. Next, sugar. It is a 5-carbon pentose sugar called ribose sugar. That is C5H1 and 10O5. 10O5, that is right. So phosphate, it is also having a phosphate. It is in the form of orthophosphoric acid. Orthophosphoric acid, H3PO4. So the three very important nitrogenous, the chemical composition of RNA is what? The nitrogenous bases. 2 are purines and 2 are pyrimidines. What are purines here? In RNA, it is adenine and guanine, and pyrimidines are cytosine and uracil. With respect to sugar, pentose sugar, that is the 5-carbon sugar. Here it is ribose sugar. And it is also having a phosphate group, hence the name nucleic acid, right? So it is having H3PO4. This is, and what will be the nucleosides of RNA? Adenine plus ribose sugar forms adenosine. Guanine plus ribose sugar form guanine. Cytosine plus ribose sugar form cytidine. Uracil plus ribose sugar forms uridine. There it was thymidine. In DNA it was thymine. Thymine plus deoxyribose sugar. Deoxyribose sugar. In case of DNA. See the difference here. What are the nuclei? What is nucleoside? Sugar plus 4 nitrogenous bases forms the sugar plus 4 nitrogenous bases forms a nucleoside. Nucleoside plus H3PO4 forms nucleotide. As all this you have studied even in first PUC and also have explained in DNA. What is the difference between a nucleoside and a nucleotide? Nucleoside is a combination of sugar. In DNA it is deoxyribose sugar plus 4 nitrogenous bases, AGCT. In case of RNA, the sugar is ribose sugar plus 4 nitrogenous bases, that is AGCU. And what is nucleotide? Nucleoside plus phosphoric acid forms a nucleotide. Here also nucleoside plus phosphoric acid H3PO4 forms nucleotide. So what are the nucleosides of RNA? Adenine and ribose sugar plus ribose sugar forms adenosine. Guanine plus ribose sugar forms guanine. Cytosine plus ribose sugar forms cytidine. Then uracil plus ribose sugar form uridine. So with respect to nitrogenous bases, 2 purines and 2 pyrimidines, adenine, guanine, cytosine and uracil. Sugar, 5-carbon sugar, that is pentose sugar called ribose sugar. And 3, that is the phosphate group, that is H3PO4. So what are the nucleosides of RNA? Adenine plus ribose sugar, adenosine. Guanine plus ribose sugar, guanosine it is. It is guanosine. Not guanine. Guanine is one of the nitrogenous bases. Here it is correct. Cytidine and uridine. If you add H3PO4 for this, PO4 plus H3PO4 plus H3PO4 plus H3PO4, it is going to form a nucleotide. What is the nucleotide here? Adenylic acid. Here guanidinic acid. Here cytidinic acid. Here it is uridylic acid. Here it will be uridylic acid. Whereas in case of DNA, it will be thymidinic acid. So this is about RNA. So as I said, the nucleotides of RNA, adenosine plus phosphate forms what? Adenosine monophosphate or adenylic acid. You can also call it as AMP, then GMP, CMP, UMP or uridylic acid. So here adenosine plus phosphate forms adenosine monophosphate or adenylate acid, guanosine phosphate or guanylate acid. Then cytosine plus phosphate forms cytidine monophosphate or cytidilic acid. Here uridine plus phosphate forms uridine monophosphate or uridilic acid. Short form you can write it as AMP, adenosine monophosphate, GMP, guanosine monophosphate, CMP, cytidine monophosphate. Monophosphate means what? One phosphate group is added. So one phosphate group is added. Hence the name monophosphate. Or the other name is what? Adenylic acid, guanylate acid, cytidilic acid and uridilic acid. So these are the 4 nucleotides of RNA. So what is the first one? Adenosine phosphate, AMP or adenylic acid. Second one, guanosine plus phosphate, guanosine monophosphate or guanylic acid. 3. Cytosine plus phosphate, cytidine monophosphate or cytidilic acid. 4th, uridine plus phosphate, uridine monophosphate or uridilic acid. So these are the nucleotides of RNA. Types of RNA. So there are 3 types of RNA we come across, as we already know that non-genetic RNA. RNA is of 2 types, non-genetic RNA or cellular RNA and genetic RNA. As I already said, what is non-genetic RNA? Non-genetic RNA means what? When it is not acting as a genetic material. So when it is not acting as a genetic material, then what is its role? So it is mainly involved in protein synthesis. It is involved in protein synthesis. Then genetic RNA, it is acting as a genetic RNA. So when it is acting as non-genetic RNA, it will always be single-stranded. Whereas when it is acting as genetic RNA, it will be double-stranded. It will be double-stranded. Here when it is acting, it will be single-stranded. Right. So cellular RNA is of 3 types, namely ribosomal RNA, messenger RNA and transfer RNA. Ribosomal RNA, short form is rRNA, and mRNA 1, 2, 3. Transfer RNA, it is also called as soluble RNA, or it is also called as adapter RNA. Soluble RNA or adapter RNA. rRNA, ribosomal RNA. mRNA, messenger RNA. tRNA, transfer RNA are the 3 types of cellular RNA, or otherwise called as non-genetic RNA. Non-genetic RNA. So, what are the types of RNA? There are 2 types of RNA. Non-genetic RNA. Non-genetic RNA is also called as what? Cellular RNA. And the other one is genetic RNA. Cellular RNA is of 3 types. Ribosomal RNA, or otherwise called as rRNA. Messenger RNA, otherwise called as mRNA. Then tRNA, that is transfer RNA, which is also called soluble RNA or adapter. So the very function of all these 3 is what? Protein synthesis. And genetic RNA is always double-stranded when it is acting as a genetic material in some viruses. So ribosomal RNA or RNA, it constitutes 80% of cellular. So total RNA is made up of, that is rRNA. The very word itself will tell you, ribosomal RNA. And it is synthesized by nuclear DNA. It is single-stranded molecule but undergoes coiling at many regions. So what is the feature of ribosomal RNA? Though it has a great role to play, but much information about rRNA is not given. So it constitutes what? 80% of the total cellular RNA, and it is synthesized by nuclear DNA. It is single-stranded, as I said, because it is acting as what? Non-genetic RNA. Obviously, it will be single-stranded, but undergoes coiling at many regions. So what are the functions? What are the functions of rRNA? Forms a structural component of ribosomes. As I said, the very word itself will tell you ribosomal RNA. That means it forms the structural component of ribosomes. Recognizes and bind to mRNA during protein synthesis. So when we go for the next session, where you will understand the central dogma of protein synthesis, then you will understand what is the role played by rRNA in protein synthesis, and maintains orientation of tRNA during translation. So two very important events takes place in protein synthesis. One is called transcription and the other one is called translation. So the genetic message that is transcribed from DNA to mRNA is called transcription. From there on, mRNA passes on the genetic message to tRNA, from tRNA to rRNA, and that process is called translation, right? So ribosomal RNA or rRNA, it constitutes what? 80%. So most abundant RNA that is found in cellular RNA is rRNA, that is ribosomal RNA. And it is synthesized by nuclear DNA. As I said, it is single-stranded molecule but undergoes coiling at many regions. Functions, forms a structural component of ribosomes, recognizes and bind to mRNA during protein synthesis, maintains orientation of tRNA during translation. Moving on to Cloverleaf model. So Robert Hawley, a scientist, proposed this model for Cloverleaf for tRNA called Cloverleaf model of tRNA. According to Robert Hawley, a scientist, proposed this model for tRNA molecule, and hence it is called, because it looks like a clover leaf, hence it is called Cloverleaf model. What are the features of this Cloverleaf model then? As I said, tRNA is one of the cellular RNA which takes part in the process of protein synthesis in transferring the amino acids from the cytoplasmic pool to rRNA. From rRNA, it goes to ribosomes to make a particular protein. So the very word itself will tell you, transfer means what? The whatever genetic message is given by mRNA is taken by tRNA to pick up that particular amino acid from the cytoplasmic pool and give it to rRNA. From rRNA, it goes to ribosomes. So here the Cloverleaf model has the following features. The tRNA consists of 4 major arms. It is having 4 major arms, but one is called the short arm, or it is otherwise called the extra arm. The 3' end of the amino acid binding arm has CCA nucleotide sequence, which is the binding site for amino acid. So the Cloverleaf model has how many arms? 4 major arms, where one arm is short arm or extra arm, and the loop occurs near 3' end, and it is a ribosome binding site. It helps in binding of tRNA to the ribosome. Near the 5' end, there is DHU loop, which recognizes the enzyme amino acid acyl synthetase. Opposite to acceptor arm, there is anticodon loop with 3 complementary nucleotides that specify an amino acid. tRNA, as I said, will pick up only that specific amino acid from the cytoplasmic pool to the site of protein synthesis. As you know, ribosome is called the protein factory. So tRNA transfers the specific amino acid to the site of protein synthesis. tRNA on one hand reads the code and on the other hand binds to the specific amino acids. Therefore, it is also called as adapter molecule by Francis Crick. As I said, you can also call tRNA as soluble RNA, or you can also use the word called adapter RNA. Why it is called adapter RNA? Because tRNA on one hand reads the code on the other hand, which binds to the specific amino acid. Therefore, it is called as what? Adapter molecule by Francis Crick. So genetic RNA. Now that was all about the non-genetic RNA. Non-genetic RNA is of 3 types. We have studied rRNA, mRNA and tRNA. Now moving on to genetic RNA. As I said, when will genetic RNA, when will RNA act as a genetic material? When it is double-stranded, and acts as genetic material in some viruses. So in some viruses, it is going to act as a genetic material. So the RNA which serves as a hereditary material is called as genetic RNA. So how do we define genetic RNA? The RNA which serves as a hereditary material is called genetic RNA. It may be double, usually it is double-stranded. Single-stranded viruses like TMV, CMV. TMV is tobacco mosaic virus. CMV is cauliflower mosaic virus. HIV, human immunovirus. So double-stranded virus. So here we can classify them into double-stranded and single-stranded R. So single-stranded virus is like, even it might be some cases acting as a genetic material, even in case when it is single-stranded, is like TMV, that is tobacco mosaic virus, CMV, that is cauliflower mosaic virus, HIV, human immunovirus, which causes a dreadful disease called AIDS. Double-stranded, like in Rio viruses, then in rice dwarf viruses. So what is genetic RNA? The RNA which serves as a hereditary material is called as genetic RNA. It may be double or single-stranded. So what are the examples you are going to give for single-stranded viruses which have single-stranded RNA like TMV, CMV, HIV, etc. And double-stranded, like we have Rio viruses, rice dwarf viruses. Hope you have understood the concepts what I have explained in this particular session. All the concepts what you learn in this particular chapter called molecular basis of inheritance is very, very important. As you know, it carries a very huge marks of 10 marks. So all the concepts are very important from examination point of view. So what are we going to study in the coming session? We are, as I said, non-genetic RNA or cellular RNA is mainly involved in protein synthesis. So we'll be studying about protein synthesis, the central dogma of protein synthesis in the coming session. So I'll be back with some more interesting concepts of this chapter. Till then, goodbye and thank you.